My name is Mariana Myers, and today I will be giving a brief overview of Huntington's disease. Huntington's disease is an autosomal dominant neurodegenerative disease, meaning that if one parent has the gene for this disease, there's already a 50% chance that their offspring will too. But it's not that simple, because simply having the gene does not mean symptoms will occur. There are carriers of the disease who have a copy of the gene but remain unaffected, and there are those who are affected and have a copy of the gene as well, but they show symptoms. So, what determines if symptoms will occur? To know that, we first need to understand the genetics of Huntington's disease. On chromosome 4, locus D4S10, there tend to be a repeat of 3 nucleotides, C, A, and G. This is called a trinucleotide repeat. Now let's take a look at the number of repeats versus the outcome. A typical person has less than 30 repeats, which makes them unaffected. Between 30 to 37 repeats means there's a high chance of passing the gene to offspring. These are the carriers referenced earlier. And over 37 repeats means there's a high risk of developing Huntington's disease. So for those affected, how does Huntington's disease develop? This is called the molecular pathogenesis, or how a disease starts and progresses. DNA gets converted to RNA, which gets converted into protein, in 2 processes called transcription and translation. When the CA gene trinucleotide repeat is transcribed and translated, it encodes for an amino acid called glutamine. This becomes troublesome because a typical Huntington protein present in everyone's brains looks like this. It has a polyQ or multiple glutamine domain here. However, a mutant Huntington protein present in Huntington's disease patients looks like this. Notice the expanded polyQ domain. This expanded polyQ domain induces changes in the protein, causing it to form intracellular aggregates or large inclusions of misfolded polypeptides. These aggregates can affect many things, such as proteostasis, axonal transport, transcription, translation, and mitochondrial and synaptic function. Long story short, they cause neurodegeneration, which leads to the degeneration of the brain and symptoms associated with Huntington's disease.